in the last previous slide. So, raw like I said, qualification department in pharmaceutical involves experiments maintaining the controlled manufacturing environments occupied by the regulated process and by equipment verified specification already into the raw material and basically Superman is medium cellular noise is coming from the cellular problems. This required for the pharmaceutical product manufacturing. Pharmacopoeia, United States Pharmacopoeia and Japanese Pharmacopoeia. So those pharmacopoeia provided the specification parameters for analysis of the raw materials, the process, microbiological testing, as well as the finished product testing. So we have to follow specific pharmacopoeia standard as per the regulatory and the agency requirement. So the next is the process validation and the process. Manufacturing processes must undergo the rigorous validation practices. So whatever product we are manufacturing, first we have to validate the manufacturing process by using the different techniques and by using the guidelines. So there are three types of the process validation. One is the prospective validation, second one is the component validation, and third one is the retrospective validation. So whenever the only launch one new product, so that time you have to perform the prospective validation considering the design critical process parameters, critical quality attributes. Whatever the parameters we define or identify during process validation, those parameters are being monitored within the routine manufacturing as well as commercial manufacturing. So that helps to perform process validations initially, and this validating method should be adopted and followed for the routine manufacturing by using the master manual record of the batch manufacturing process and the process controls. So whatever identified the critical process parameters and the critical quality attributes, those parameters have been monitored during the commercialize of more batches. So, between your individual, so molecules parameters by function, supervisor as well as the computation department to achieve the consistently on the levels. So, environmental and the physical. So, how to maintain or control the environment in manufacturing. So, this control details the initial planning. It means reducing the this so HVAC system is one of the implementation and integration. It is the equipment which helps to maintain the environmental condition in the production facility. Just like to provide to maintain the temperature as well as the humidity control. Additional pressure differential between the adjacent room, it helps to prevent cross contamination between outside area. Then microbiological monitoring method is theoretically defined after designing of the HVAC system or the after facility design. Again, there EEG validation also performed. And based on the EEG validation, you have to monitor the microbiological contamination in the manufacturing area. Periodic reverse now for 15 days manually validate the fitness. So there are two types of this microbiological monitoring. One is the passive sampling and second one is the active sampling. One method is to expose the microbiological media rate in the environment to capture the microbiological contamination, identify the microbiological contamination in the specific area. The second one is by using the pH sample. So education system is maintained. It helps to control microbiological contamination as well as the particular contaminations. So how controls? So the education system is having a HEPA filters, and this size of this HEPA filter is the 0.3 micron filter size. This particles. So it helps to trap the microbiological contamination on this HEPA and supply the fresh and quality air in the manufacturing. So accordingly, so with the help of this education system and other microbiological control, so maintain the environmental condition, prevent microbiological contamination facility. Third one is the final product testing and release. So after manufacturing of the drug, by taking the required controls as per the validated procedures. So after finished product planning, so already the insurance department select the correct samples from different locations on production area, and those samples submitted to the quality control requirement, and then quality control perform the analysis as per the validated method and as per the pharmacopoeia standard perform and to identification of particular drug, unity of drug, potency, impurity profile, is sterility testing is required only for the injectable products, these large volume parameters of small volume parameters. So after getting the satisfactory results against the pharmacopoeia standard, we take those parameters and release the product for the distribution in the market. So these are the mechanisms for issuing the product quality flow from receipt of raw material, qualification of raw material, the distribution of product and final market suppliers. So please go to the next slide. Yeah. So compliance and data integrity. So quality consideration within the fifth slide is the fifth slide. So compliance of data integrity principles. So the pharmaceuticals involve consistently adhering to the regulatory requirements. So for maintaining the documentation in the manufacturing area as well as the quality control and the qualification department. So based on the guidelines, we have to follow the good documentation practices. So as per the regulatory requirements, internal policies and documented procedures. So that is one of the most critical parameters for the compliance and maintaining the data integrity. So as the quality system detects the accurate and reliable records. So control principle needs to be followed for the recording materials and maintenance of the documentation. So control is called so A stand for the data should be attributable, usable, contemporaneous, original, accurate, then plus means what? The complete, consistent, memory and available. So all these parameters should be followed while recording and maintaining the data. Means attributable, visible, data should be visible, then contemporaneous, whatever the details we see it is a PLC or any lab or any instrument, same business shall be recorded to the logbooks or any test data sets without any falsifications. Then original data should be recorded, accuracy should be maintained, and complete data should be recorded. So incomplete data it means we cannot investigate only market computation from market compute or the requirement incomplete requirement will during the analysis of during the evaluation of many products. So these are the principles that we follow in the pharmaceutical industries. So continuous CFR compliance will be maintained. So data from the laboratories and audited will be provided and control access and electronic signatures shall be provided to the respective area. It helps to provide the manipulation in this data and appropriately input those data before using the decision making during the use of the batches. So electronic data must compare the manual CMR 5 guideline is provided by the USFDA. Then European EU GMP guidelines are provided the analysis relevant for maintaining this electronic data storage, and it helps to control access and electronic signatures. Compliance also extend to audit preparedness, regulatory submissions, and so all these data monitoring, so separate handling of documentations, security, so how handles the data, how to control this data, what is the relation period of this data. So all these things should be recorded in the standard operating procedure guidelines. Whether it has those common procedures should be followed. Executing your department training to a particular and concerned department. And manufacturing area, storage area, political. So audit and training important. So companies must conduct the routine internal audits, gap assessment, then staff training programs periodically training should be reported to the concerned department, whatever they should be. They have respective departments. It helps to reduce the regulatory needs and ready for the regulatory needs. So ultimately, the strong compliance also will be maintained. So it helps strong compliance ensure that the quality system operates effectively and the patient does not pharmaceutical product. So material compliance ensures clinical data falsification and ensure the patient does not products. So please switch to the next slide. So quality consideration in the medicinal industry. So what is the actually quality contribution and quality consideration in the medicinal industry? So give a whole thing. So the example of the molecules. So the contribution in the medicinal industry, they also involve anticipating the and controlling the quality risk associated with the chemical synthesis and drug design. So while the drug design, there are a number of studies to be performed during the drug design, and this activity relationship to enhance the potency, selectivity, and safety considerations. So that time while you are designing of these products, you have to perform the various study, stability study, then what is the risk while maintaining this formulation or using those products formulations. So all these parameters shall be studied in the designing of molecules. That's why the molecular considerations during the medicinal chemistry while the molecule is developing. So, integrated profiling Papers are analytical techniques such as HPLC, then GCMS, so gas chromatography and mass spectrophotometry, then liquid chromatography and mass spectrophotometry, then higher spectroscopy to identify the known and unknown impurities. So, using this designing of this drug design, so we have performed the impurity profiling test. So that is the role of quality assurance to investigate and select impurity profiling in the molecular development as well as using the composition statements. So understand the reaction of the intermediates while identifying this impurity profile. And according to the polymorphism studies, ensure that the crystallizations forms during stable and storage, different devices papers, those parameters have been monitored during the static studies. So according to we can design or develop the shape of molecules, what are the shape like how many requirements of the products and generation of the utility. So all these parameters, critical parameters should be monitored during the stability studies. So next slide is
Next slide please.
8 minutes. So the operating system rules and operation of the PhD pharmaceutical industry. There are number of rules you have to maintain and follow for maintaining the good manufacturing practices in the pharmaceutical industries. Like variations, process qualifications, then quality management, duration management, change management, right? Corrective and preventive actions and operational parameters in manufacturing, includes manufacturing integration, includes parameter to be monitored during the manufacturing. So this next slide, that is the rules. Those rules we have follow and maintain as per the regulatory requirements and guidelines provided by the national agencies or authorities. So variations, qualification and the risk management. So variations, there are a number of variations like process variations, then equipment qualifications, lead variations, then prototype studies. So all of these activities needs to be maintained and performed or executed in the pharmaceutical industries for producing the quality and safe product. So first the equipment qualifications. So initially, if we want to procure one equipment area for manufacturing area, so first we have to prepare the user requirements specifications. First we have to prepare the requirements. What is the requirement capacity? What is the MSC of this product? What product we are manufacturing in this equipment? So all these parameters first to capture in the device accordingly. So vendor provide the design configuration. So after review this design configuration approved by our side, then we go for the procurement and after procurement of those equipment. So then install in our facility and perform the installation configuration. Then verify the operational parameters during the operational configurations. So operating is respect to operational parameters, then we prepare the standard operating procedure in the draft stage. So after preparing this, after completing this operational qualification, then we go for the performance qualifications. It is to be performed by validate and verify the functional parameters. If we get the proper functional parameters as per our requirement, then we qualify those equipment for the usage the routine basis. So routine validation of product and perform the process validation. These are the equipment qualifications. These are the different stages. Design qualifications, installation qualification, operational qualification, and performance qualifications. After satisfactory performance qualification, those equipments hand over to the production for routine in this. So after satisfactory qualification, then we go for the process validation. So process validation demonstrates that manufacturing process consistently yield products meeting the predefined specification and quality requirements. So that is the process validation. So based on this process validation, we validate the process and same process based on the validation, we prepare the corresponding HTTP report approved by the validation state, and after this approval, same copy issued to the production for the execution of manufacturing batches. Then cleaning variations, the cleaning variation, it ensures that the residue from the previous product or the cleaning previous product residues or the cleaning agent whatever we use for the cleaning of this equipment do not cross contaminate with the subsequent batches or major batches. So that's how we have to perform and execute the cleaning variations by defining different techniques, chemical analysis as well as the microbiological contamination monitoring. So based on that, we validate the cleaning and same cleaning procedure should be adopted during the product changeover. This is the cleaning management system. Connects the analytical with user validations. Whatever the product we are testing in the particular department, so we have to follow the pharmacopoeia standard as well as if we follow the pharmacopoeia standard but before adopting the particular development for this method, first we have to validate by ensuring the validated procedure to accuracy of these method decisions, their reproducibility, their awareness of this method. So we have to follow those parameters and validate them with our scientific validation to validate this method. Then say in the reference of this document, the validation department again prepare the each product specifications and say approved by validation state, say method will be submitted to the quality control department for performing the analysis of specific from each product and raw material. So these are the validation standards. And last is the quality risk management. So as I said, as per the ISH Q9 guidelines, so they are providing the quality risk management. So QRM is a system, this structure system. It helps to identify the risk, it analyze and mitigate those risks to minimize the risk of during minimize the risk of our product during the validation. There are a number of tools which we have to follow as per their nature of the identified risk. So, one of the tools is the FEE, then quality analysis, hazard analysis, hazard analysis in critical control parameters. So, usually the companies are identified, assess, and mitigate the potential risk. So, failure more effective analysis. So, these are the tools which we have to follow for quality risk management and to minimize the risk for the product safety and product quality into the pharmaceutical industry. Let's go to the next slide. So apart from that, so these are the quality management tools which we are going to maintain in the pharmaceutical industries. Whenever we instead of software or quality control department or any department, so deviation management process should be followed. So deviation management means whatever the incident happen in any department within the manufacturing or within the handling of matter. So immediately initiate the deviation form, then investigate those incidents by using the different tools to identify the probable root causes of the root cause. So there are a number of tools for the investigation of the deviation, like five tools, ask the questions against why. So this is number five. We get the actual probable root cause of the particular incident. Then the second one is the fishbone diagram, Ishikawa diagram. So if there are multiple process of those instances of any compromises. So we have to follow this visual diagram or system. So there are number of routes we have to follow for the investigation and identify the proper root cause of the particular disease. So investigation then identify the root cause, and for mitigation of this root cause, then we have to define the purpose, corrective actions and the preventive actions to avoid such type of preventing such type of incidences in future. So those the deviation management system is in place. It means the quality system is several quality systems available in the pharmaceutical industry. Remain the deviation or any abnormals into the pharmaceutical industry. So this is the deviation management process. So mitigation of this non-organized deviations. So in kappa implementations. So whatever the design or defined kappa or mitigation of this risk. So accordingly, kappa implemented prevent the actions to eliminate patient time, reduce the recurrences, this in future. The manufacturing activities. So each and every activity shall be ensured by the quality assurance department. For example, whatever the raw material is in the store department, you see analyze those raw material and the clearance to the store department for the issuance of those raw material to the production of the product management. Before hand over this raw material to the production department, first the quality control ensure and assure those raw materials, investment those raw materials are used or not. If after getting the satisfactory results of those raw materials are released, we will must ensure and use the line tellers for those raw materials and over to the production. So each and every stage in production as well as the quality store department, every degree in process qualifications process ensures and verify all the parameters giving the line tellers as per the checklist provided, and we use the line tellers to start directly for production. So each and every activity shall be monitored by the qualification department with the languages throughout the manufacturing process. And after final release, before giving the final release, we also with the quality control. And after getting satisfactory individual as well as the cash and executive reports, all by reviewing the evaluation systems or evaluations which was recorded during this manufacturing. So all the parameters reviewed by the executive after getting satisfactory reports, then after that the executive release those products for the distribution and for the market. Okay, that is the main role of the correlation department. So go to the next slide. Final activity and integration is summary. Next slide. So basically, so we already discussed the role of correlation in pharmaceutical and ensures that the medicines are safe, effective, and complete with the science and regulations and control over the products life cycles. Basically, promoter with respect and the market surveillance. And also the contribution of the correlations to medicinal chemistry to evaluate the structure of molecule synthesis complete process, then integrated profiling, identify the impurities, then post regulatory study, then stability studies, and then designing and development, and after development, so again same drug is to be used for the formulation of any process like tablets, capsules, injectable, liquid, all those integration unit. So quality assurance contribute to the integration of the medicinal chemistry base, quality, then reduce the risk, support the regulatory successes, means is directly open the market after getting the certification from any authorities. So automatically open the market for the product for the country and for the patient defense. So that's what is the main role of quality assurance department in the pharmaceutical industry. It ensures and compliance with products. So then, so what is
So any questions, audience? Okay, sir. Thank you, sir. Thank you. Yes, sir. Yes, sir. Yes, sir. Thank you, sir. Thank you, sir. Thank you for your sir. Please, sir. You will see me, sir. Please, sir.
Respected dean madam, my colleague faculty and dear students, good afternoon, one and all. So I am very glad to be a part of today's speaker to this national conference. And to this national conference, my topic is PROTACs, a new paradigm in anti-cancer drug discovery. The introduction of this PROTACs has brought a transition in the cancer treatment. So what are PROTACs? The abbreviation of PROTACs is proteolysis targeting chimeras. So proteolysis targeting chimeras. Proteolysis is a natural process that occurs in our body. That is, the unwanted excess protein produced by the cells will get destroyed. So chimeras are the structural moieties. They are referred to be the hybrid molecules. They are generally formed physically by binding the two different functional groups. So how why the products are calling as chimeras, we will get to know in the next slides. So from blocking to destroying. So we know the traditional anti-cancer drugs will act by inhibiting mode, like lock and key. Okay, the drug will find a specific site on the protein on the cancer cell and it will inhibit. So it works through lock and key. By coming to the products, it works through the destroying mode. It drags the protein. It grabs the protein completely and it will it will drag into the natural destroying phenomenon. So instead of blocking the protein, it is completely destroying the protein. So what are the limitations of traditional inhibitors? So usually using conventional anti-cancer drugs will work with the inhibiting mode. They are having some disadvantages like the single binding site. So the protein will have a single binding site. If the drug finds that binding site, then it will bind to the protein and it will inhibit. If it doesn't find that it doesn't inhibit. So this is one of the disadvantages. The secondly, the cancerous cells will be under continuous mutation mode. If once the cancerous protein has been modified or mutated, then again our drug cannot identify and it will not inhibit it. The third one is high occupancy is required. That means more amount of the drug is needed for inhibiting the protein, for inhibiting the cancerous cell. If more amount of the drug is used, it leads to the toxicity. So these are the limitations of traditional inhibitors. So next, beyond the reach of conventional drugs. So how products are acting beyond the conventional drugs? So many cancerous carbon proteins have completely smooth shallow surfaces. They are having smooth shallow surfaces. They are not having any pockets. Any pockets or sites. So it is difficult for the traditional drugs to identify the protein, to identify them and to inhibit it. Instead of it, how these drugs are acting, product drugs, it grab the protein completely. Even though it is having a shallow surface or smooth surface, it will grab the protein and it will drag into the natural garbage system. That means complete protein is inhibiting. Okay, next, no active site is needed. So for this, as it is acting in destroying mode, it doesn't need any active site. So this is the product architecture, the structure of a product. A product is a bifunctional molecule which consists of three modular components present in it. It contains a target protein which binds to an E3 enzyme ligase and also an anchor ligand. Both these anchor ligand and sorbate ligand are connected with an linker. So these three are the modular components present in the product. So once this three will get assembled, the enzyme ligase taps the target with the polyubiquitin chains which signals to the 26 proteasome to permanently degrade the cancerous cell. So once that taps into that system, the product again it detaches unharmed and this cycle is a repeated one. See here the three modular building blocks. The anchor it consists of the product consists of an anchor. That anchor it binds to the E3 ligase. So this is a E3 enzyme 3 ligase. The second one is the warhead. This warhead will bind to the target cancerous protein. The third one is the linker. So here anchor it docks into the E3 ligase pockets. And the warhead it recognizes the target protein and these two will get binded with that linker. See here the proteolysis chimeras are bifunctional molecules that acts as molecular bridges. One end binds the target oncogene and the other recruits an E3 ubiquitin ligase. So once it recruits to the equilibrium ligase, it was modified and it leads to the it grabs into the natural disposal system. So this is the natural cellular degradation pathway that occurs in our body. So here it consists of three enzymes. E1 enzyme activates the ubiquitin, E2 transfers the ubiquitin, and E3 ligase attaches this ubiquitin to the cancerous protein. Later, it is polyubiquitinated protein enters to the 26S protease where the protein will get degraded. See in the image you can see E1 is activating it and E2 is transferring and E3 binds this ubiquitin with the target molecule and this target molecule will be forwarded to polyubiquitinated and later it entered to the 26X protease where the degradation of protein has been done. So the most pharmacological problem we have seen with this product is the hook effect. So we are saying this is a three-modular in which one is bind to the target protein and another is binding to the so instead of binding instead of producing this ternary mixture here the excessively high concentration of product is added, the individual product molecule will saturate. Instead of forming a ternary mixture, it is forming a binary mixture. So it has to bind to ligase enzyme, E3 enzyme, and also it has to bind into the protein of interest. It has to bind to protein of interest and also it has to bind to the E3 ligase enzyme. So instead of binding to the three forming of and ternary complex, it is producing binary complexes like protein of interest, protan and protan E3 ligase. So this the formation of these binary complexes will decreases the degradation efficiency. In order to avoid this binary functions, we will use a neo contacts. So using these neo contacts will stabilize the protein-protein interactions between the protein of interest and E3 ligases. So the protein linkers plays a very major role because the length, chemistry and the flexibility of protein linker is very important. So if the linker is too short, the severe steric interactions will be occurred and it will push the E3 ligase and the protein of interest apart. So rendering ternary assembly impossible. If the linker is too long, it will fail to bring the proteins close to a fellow efficient ubiquitin transfer. So it dramatically increases the molecular weight, rendering the compound completely incapable of crossing the cell membranes. Okay, so this is about product linkers. So here there are some product linkers we are using: the alkyl chains, polyethylene glycol, saturated heterocyclines, and alkynes and triazoles. So these can be used as a linker. The most commonly used are the alkyl chains and PEG polyethylene glycol. So coming to the mechanical strengths, alkyl chain and polyethylene glycol flexible in nature and mostly they are used. So which provides structural elasticity and they are highly lipophilic. So next, saturated heterocyclic, they are rigid in nature. It locks the bridge in a Z shape. So these are less used. This fiberazine, fiberidine and alkyl and triazoles are less used. So next, taking the concept of these products, the researchers has developed the new smart products. This is one of the next generation product, the hypoxia activated product drugs. So hypoxia is oxygen, in absence of oxygen. So these product drugs will be activated in the absence of oxygen. Cancerous cells are deprived of oxygen, whereas healthy cells contain oxygen rich. Okay, so these cancers attached directly to the EP angle. In normal oxygen-rich healthy tissues, the molecule remains completely inactive. When it enters, it will be remain inactive. However, once the drug enters the hypoxic microenvironment, the local enzymes cut off the shield. It will releases the potent drug and selectively destroys the target protein. Next, clean chemistry assembly. Some researchers have developed this product by using clean chemistry. So they joined the three modular components with the help of the clean chemistry. See here, the anchors, the variable linkers, markets, they connected directly in the aqueous solutions and they have given perfect yields. Next, linkage-free designs to create a weight and size. So, as it is containing three molecular, the molecular weight of this is very high. In order to reduce the molecular weight, some researchers have prepared these linkage-free designs. They directly join this bond head with the angle with a single bond or with a single sequence, which reduces the molecular weight. Next, macrocyclic products. So, by wrapping the secondary macrocyclic ring around an existing flexible product structure, they can physically freeze the compound. The compound will get freezed in its active conformation. So, this structural locking mechanism improves cellular potency and enhances selectivity towards the specific protein divisions. Next, these are the advantages. Destroying the undruggable, that means most of the cancerous proteins will not have the active sites. So 80% of the proteins will not have the active sites. Those proteins are treated as undruggable proteins. So here, these products will destroy these undruggable proteins also. Okay. Instead of finding at the site, they will grab the protein and they will grab into the natural system. The second one, conquering drug resistance. This is also as some of the cancerous cells are in the continuous mutations. These normal drugs cannot find them. Here, the products completely shred the entire target architecture. They retain full efficacy in this heavily mutated, block-resistant variants of enzymes. So, they will destroy the mutated ones also. Isoform selectivity. So, a drug primary molecule from the alcoholistic, it achieves selective destruction. That means here similar isoform cells can also be targeted by this products. So next, event driven catalytic machinery. So standard inhibitors requires a constant high dose to keep an active site cut. So a single catalytic product molecule repeats its shedding cycle over and over. So in the normal traditional inhibitors, we have to take more amount of the drug, which leads to the toxicity. Whereas here, the small amount of drug is enough, so it will repeat its shedding cycle over and over. This translates to the incredible low patient doses. So this is clinical proof of products. The phase 1 has been started in the 2019 and now we were in the phase 4. So multiple programs has been done and it is successful model. So the drug also has been released into the market. So some of the products are in the clinical trials. The companies like Arvinas, Pfizer are sponsoring for these drugs. So these drugs are targeting these enzymes, estrogen receptor, androgen. So and they are in these particular phases. So the formulation challenges include, so it is very difficult for the oral food solubility and the oral absorption for since because the molecular weight is very high. The advanced delivery systems like nanoparticles, antibody conjugates are under active investigation. So cancer-specific targeting strategies aim to concentrate products at the tumor sites. Optimizing nucleic selection for tissue compatibility is a critical translational focus. So this is Bepano is the first drug approved on May 1st, 2026. So it is available with the brand name Bepano. The drug is Webdigest jointly developed by the Alminas and Pfizer. So this is the first ever FDA approved product. So this is the first approved product. So along with this drug, the FDA also approved a test. So the test name is authorized quadrant 360 CDX test. So the persons who are enrolling to take must undergo this test to detect the ESR1 mutations because this drug is very much effective in the treatment of this ESR1 mutated advanced or metastatic breast cancer patients. So it should not be given to any patients if they were in the first phase or the second phase. It should be given to the patient who were mutated with this ESR1. For this only, the drug who are taking the patient who are taking this drug must be diagnosed with this test. Then only they are allowed to take this drug. So here first companion diagnostic will be typically done with a simple liquid biopsy or blood test. So the second one, the target identification. So the oncologist must confirm that tumor expresses the specific or mutated protein. So once the doctor has to confirm that the patient is diagnosed with that mutated protein, then only the product was given. Okay. So the mechanism of action of this drug weapon. So here it is a molecular bridge with two ends. One end binds to the estrogen receptor on the cancer cell, and the other binds to the E350 ligase. So by bringing these two together, it tricks the cell to attach ubiquitin tags to the research receptor. Later, it will be flanked onto the cell's proteasome, which acts as the protein disposal system, which completely deploys or destroys the estrogen receptor. So the common side effects were reported fatigue, mild nausea, and musculoskeletal joint pain. It is usually managed at home with supportive care, minor dose adjustments. So special cardiac monitoring should be done if we were taking these products. So these were the advanced clinical trial products, so which were under clinical trial. Galantamide, ARP-766 and GP209. So it is glutamide. It is an oral product for metastatic castration-resistant prostate cancer. It is selectively destroys the androgen receptor. So the second one is the next generation androgen receptor degrader evaluated for advanced prostate cancer. The third one is developed by pharmaceutical. It is a unique topical product applied to skin to treat the androgenetic alopecia. Thank you.
Thank you so much, Kumar, for your wonderful speech by you. On behalf of organization, I would like to express a sincere gratitude to our beloved email, our esteemed faculties, speakers, delivers such an insightful and informative session today. I also like to thank the organizer, faculty members, participants, everyone who have contributed to make the program a successful event. Thank you all. Thank you all.